Okay, hi everybody. Um, so I hope you enjoyed the tutorials that Chirag ran a few days ago. Today I'm going to focus, kind of build on that theme. Hopefully you understand how convolutional neural networks operate and how they can be used to map DNA sequences to transcription factor binding data. So today I'm going to give more of a research talk, sort of building on that theme. And focusing on two components. The first is using these models not just as black box predictors, but trying to understand what they're learning. That's a piece that I don't think the tutorial focused on, neither did the lecture. So hopefully you will see today how we can open up the black box and try to use it to understand the underlying syntax of regulatory DNA. And then we'll also use the models to try to understand genetic variation. Okay. And again, you can feel free to stop me if you have any questions. So I'm just going to quickly, since it's been a few days since that lecture, I'm just going to very quickly flash through 5 slides that you've seen before. Just to remind you again, our goal is to try to understand the molecular function of parts of the genome, either genetic variants or specific DNA words or syntactic units. And we're focusing on regulatory DNA and regulatory elements. These are pieces of DNA whose role is to regulate gene expression. These elements are typically bound by DNA binding proteins called transcription factors that recognize specific kinds of sequences, or I would say a range of sequences. So they have affinity for specific families of sequences. But these transcription factors rarely operate independently. They typically bind in a combinatorial fashion to complex syntax encoded in the sequence. And this syntax often has there's an evolutionary constraint to them, and it also has interesting effects on binding of the different proteins and effects on gene expression. And we talked about how we can use sequencing experiments, sequencing-based experiments, functional genomics experiments to map where these proteins bind. So here's an example of a ChIP-seq experiment for this protein. Gives you a genome-wide profile of read counts across the entire genome. So these pileups of reads where you see spikes indicate likely positions of binding. You can also use them to study other kinds of modifications to chromatin or DNA. And we also talked about how interpreting genome sequence actually is highly context dependent, because all the cell types in tissues in an individual's body largely contain the same genome. So the genome is differentially interpreted in different cell types in different contexts. And several large projects have been generating these kinds of data, genome-wide profiles. So across the entire genome, hundreds of different kinds of molecular readouts in thousands of different cell types, tissues, individuals. And so, as I mentioned at the start of the talk, we're going to focus on how we can use deep learning models to take these kinds of data sets and try to decipher two things. First is how is the DNA sequence encoding this kind of molecular activity, and then also using the models as in silico oracles, we can make in silico mutations and predict effects of genetic variants, and again try to understand which genetic variants are functional, what kinds of impacts they have, what kind of sequence syntax are they disrupting, and so forth. See if we can move my video a little bit. So let's focus on the first piece, which is trying to decipher genome syntax. So again, to remind you, what we're trying to do is take these regulatory elements and look at their molecular activity in some cell type of interest. So for example, you could take a transcription factor ChIP-seq data set, which gives you genome-wide protein DNA binding maps for different proteins. You could focus on one such experiment, and you can take any single element right here. This is a binding site for GAVP. And you might be interested in trying to answer the question in that particular binding location for the protein, what in the sequence is driving that binding event, right? And when I use the word syntax, I'm referring to not just individual words or motifs in the sequence, but also complex arrangements, like how does the orientation of this motif or this motif matter? How does the spacing between them matter? Do all three motifs act additively? Do they act super additively, multiplicatively? These are the kinds of questions we want to answer, not simply decipher individual features in the sequence. We do want to do that too, but go go slightly higher than that. Okay. And I mentioned that you can transform this into a simple machine learning problem where you're given the genome and you bin it into chunks. Let's say 100 or 1,000 base pairs. The data gives you effectively a label on every bin in the genome, and so you end up getting millions of sequences. And your goal is to learn a machine learning model that tries to transform or map these sequences across a genome to the molecular labels, which are either binary or they can be continuous. But in this case, the sequence is being mapped to a single scalar variable, output variable, which is either binary or continuous, right? We talked about, even in the tutorial, we talked about these kinds of binary models that will take sequences and input and try to transform them into a binary label or probability of the label being positive or negative, that is, a TF is bound or not. And we discussed convolutional neural network architecture, where you have layers of convolutional filters. These convolutional filters act like motif detectors. The act of convolution is the same as scanning a motif across the sequence. You have multiple filters, each of which try to learn a different pattern or motif. And the output of each convolution layer is fed to the next convolution layer. And that convolution layer builds on the features learned by the previous layer, right? So all these layers in the neural network, they're just pattern detectors. And your goal is to learn combinatorially more complex patterns as you go up the layers. And so a neural network can in a sense learn arbitrarily complex patterns, right? They can learn simple motifs in the first layer. They can learn combinations of motifs in the second. And if you add enough capacity, they could potentially learn all kinds of things ranging from orientations, affinity, syntax, spacing, higher order combinations. All of that could be learned by multiple layers of the neural network. But in this talk, I'm actually going to switch the architecture. I'm not going to focus on the binary architecture, where you take a sequence and you try to predict a single value associated with that entire sequence. Instead, we're going to move into what we refer to as ultra high resolution, in fact, the highest possible resolution, base resolution models. And I'll explain what that means. And just to motivate why that is interesting is because if you take any of these experiments that profile binding of a protein like ChIP-seq or ChIP-exo, which is sort of a higher resolution version of that, which I'll talk about today. Or there are other kinds of experiments like DNA-seq and ATAC-seq, and I'll talk a little bit about these as well. ChIP-seq and ChIP-exo, they are experiments targeting a specific protein of interest. So if you're interested in the binding sites of a specific transcription factor of protein, you will typically do a ChIP-seq or ChIP-exo experiment. And the readout you get across a genome will give you effectively the probability of or the occupancy of that specific protein across a genome. So if you need to do this for every protein, if you want to get binding landscapes of every protein in the genome, you need to do hundreds of ChIP-seq experiments. Instead, you can do a DNA-seq or ATAC-seq experiment. This experiment doesn't tell you which transcription factor binds at which position in the genome. It tells you whether any transcription factor potentially binds at a position in the genome. And the trick is simple. It uses this enzyme called DNase-1. And what this enzyme does is wherever you have proteins, any protein binding, which pushes these kind of nucleosomes away. Nucleosomes are like structural, protein complexes that DNA is wrapped around. So when a protein like a transcription factor binds a DNA, it typically moves these nucleosomes apart and creates what's called open chromatin. And this DNase one enzyme can go and basically preferentially cut the DNA in these regions of open chromatin where any protein is bound. And so what you end up getting is pileups of sequencing reads where any protein binds. And you get these kind of beautiful footprints. So wherever the protein is bound, it protects the DNA. And then this DNase enzyme will cut on the flanks of the binding sites and give you these strong spikes in the signal. But the key point to notice here is that the ChIP-seq experiment, as well as the DNA-seq or the attack-seq experiment, which uses different enzymatic strategy to kind of do the same thing. If you look at any of these profiles, these read coverage profiles, read pileups from the sequencing experiment at single base pair resolution, right? Not at like 100 base pair resolution. You don't just look at the number of reads or the signal level within 100 base pairs, but at every base pair, if you look at how many reads, what's the signal you're getting at every base pair, you often see these really kind of beautiful shapes, okay? So where the protein is bound, you kind of see this beautiful asymmetry of reads on the plus and minus strand. Here you can see ChIP exo. You can see this kind of really kind of high resolution profile flanking the exact binding site. When you have a protein bound in DNA-seq, you typically see these kinds of footprints where there's like a valley of signal right where the protein is bound, and there's a very specific shape of DNA's reads that come out of the experiment. And all of these shapes of these read coverage profiles of the signal at single base pair resolution, they have really exquisite information about the protein DNA contacts, right? So if you take this kind of beautiful profile and you just convert it into one number, right? I just sort of add the signal in this 100 base pairs or 1,000 base pairs, and I just convert it into a single number, 20, 23, 50, whatever it is. I'm trying to lose all this information, that is really capturing precisely how the protein contacts the DNA, and it might give you much more interesting information about how the sequence is actually driving binding, right? So we hypothesize that it might be very useful to, not summarize this data into a single scalar, and let's just try to model the data at single base pair resolution as it comes out, of the experiment, right? So no approximations, nothing. We're literally counting sequencing reads at every base in the genome, and we're going to try to model the data at single base pair resolution, okay? So this is the sort of the modified convolutional neural network architecture. Again, it's okay. So just to be clear, I tilted the slide or the architecture so that I can fit it better on kind of a rectangular slide. So let's just go back here. Here, this, the sequence is right here, and you're adding these convolutional filters to take it to the label, right? Now imagine just tilting this architecture 90 degrees. So now the sequence is sort of vertical, right? And that's what we're seeing right here. Again, the input to the model is again sequence. It's one hot encoded, as we mentioned before. So ACGD, ones and zeros, depending on which base is at which position. We're going to use 2000 base pair input windows, and we're just going to tile the genome with 2000 base pair windows. And we're again going to build a simple convolutional neural network. Again, you have convolutions. You have a special type of convolution here called dilated convolutions. I'll talk about this in a second. But this neural network is quite deep. It has 9 layers, as you can see. It has a classical convolutional layer at the beginning, and then it has these special dilated convolutions with what are called residual connections. I won't focus too much on this. If you have any questions, you can ask me at the end of the talk. But the key idea here is that you have 9 layers of convolutions, and what these convolutions are going to try to do again is learn patterns in the model, right? Patterns from the sequence. But now the output of the model is not just one number, right? So originally, this part of the output didn't exist in the previous model. You had convolutions and blah, blah, blah. And then in the end, you ended up with a single output value, right? Just one scalar output. So that still exists in this model. That's this part, this branch of the output. So this branch of the output is exactly very similar to the previous model. It takes the sequence and pushes it through all these convolutional filters. There's a final dense layer at the final, in the final layer that agglomerates information across positions and across filters. and then produces one scalar. And that scalar is you're trying to predict the log of the total signal, the total read counts across the entire sequence. Okay, so that's exactly like the previous model we saw. But there's a difference. There is another output head. Okay, so there's not just one output head. There are two output heads. One output head is trying to predict the total counts, as we saw before, the log of the total signal. And the other head is trying to predict, given this total counts that you predicted, how are these counts distributed across the entire 1,000 base pairs? Okay, so think of this as like a probability distribution over 1,000 positions. Literally, it's a probability at every position. If you sum the probabilities, they would sum to 1, right? So it's actually a probability distribution. And what the probability distribution is telling you is if I had, 2 to the 7 reads, right? This is log base to 7.52 log base to read counts. If I multiplied those counts by the probabilities at every position, I would get the reads at each position. Okay, so it's kind of breaking up this observed signal, which is a count of sequencing reads at every position in 1,000 base pairs, into two pieces. One is the total number of reads over the entire 1,000 base pairs, and the other is how those reads are distributed. What's the probability of observing a read at every position in 1,000 base pairs? Okay, so if you combine these two, you can effectively get the reads at every position, because you just multiply the total read counts, take the log, invert it, right? So 2 to the 7.52, and then you take that number and you multiply it by the inferred probability at every position, and that would give you the read counts at every position. Okay. Now, previously we discussed loss functions, right? Loss functions are used to optimize the model and learn the parameters of the model. We had discussed two types of loss functions, right? We had discussed a binary loss function, a logistic loss, and we had discussed a mean squared error loss. And the mean squared error loss is typically used for continuous outputs. So because this is a continuous output, we use a mean squared error loss on the log of the counts. But now this is a kind of a different kind of output, right? It's not binary and it's not a single scalar either. It is literally a probability distribution over 1,000 bases. So here what we do is we use, a nice fit for this is a multinomial distribution. And I'm sure many of you have heard of the multinomial distribution. It's like, imagine that each position in the sequence is like a bag, and the number of reads that you're getting at each position is like a bunch of balls that you're throwing into the bags, right? So each bag, each position in the sequence is like a bag. There's a probability of having a certain number of reads or balls in that bag, and then you have total number of balls that you're throwing into this collection of bags, and then different number of balls are going into different bags, right? That's sort of the classic multinomial distribution. So we can use this multinomial distribution. Our goal is to infer the probabilities. The goal is to learn or predict these probabilities. But what's going to happen is we're going to model it as a multinomial where you actually have a total number of read counts, and you have inferred probabilities, and from that you get inferred counts, right? So you can assume that the data from this output head follows a multinomial distribution, and you can use the log likelihood of that multinomial distribution as your loss function, or the negative log likelihood. Okay, so you have two loss functions now playing together. You have a loss function operating on this count head, what we call the count head. That's the mean squared error loss. And then there is an additional loss. Literally, it's an additive loss. This is one loss function. And then there is another loss function, which is the negative log likelihood of the multinomial distribution. Okay, so it's two loss functions playing together, and the model is jointly optimizing both of those losses to try to predict the total signal as accurately as possible, and then to also predict the probabilities at every position as accurately as possible to maximize the likelihood of that multinomial distribution. Okay, so when we do that, if we fit this model across some training chromosomes and we predict on test chromosomes, here I'm showing you 4 different proteins. So these are 4 different models. Each of them is trained on the same sequence input, but obviously they're 4 different proteins and they bind different positions. They have different kinds of profiles. And so it's 4 different models, and each of those models is making predictions on the test chromosomes, right? So you can see quite nicely at single base pair resolution, these models are quite incredibly accurate. Like you can, in some cases, you cannot make out the difference really between the observed and the predicted signal. And all these 4 proteins, these are very famous proteins, OCT4, SOX2, NANOG, and KLF4. They're called the Yamanaka factors, reprogramming factors. If you sort of overexpress these, at least these 3 transcription factors in, for example, skin cells or fibroblasts, you can reprogram them into stem cells, right? iPSCs. So they're very famous transcription factors in embryonic stem cells. And so, our collaborator Julia Zeitlinger did really beautiful, these are chip exo experiments. And we trained the model on those chip exo experiments, single base pair resolution. And here what you're seeing is the predictions of 4 different models for 2 different enhancers in the genome, 2 different regulatory elements. So at this element, you can see that all 4 transcription factors show different kinds of profiles. You can see OCT4 and SOX2 show very nice, sharp binding profiles. NANOG shows like a kind of a fuzzy profile. KLF4 shows also kind of a fuzzy profile. And you can see the model makes pretty nice predictions. They kind of match up quite nicely within this region. Here's a separate region in the genome. Now you see that OCT4 is also showing a little bit of a fuzzy profile. And so you can see the predictions of the model could really quite nicely mimic what's happening in the observed data. And this is on chromosomes that the model has never seen in training, right? So it's just using the sequence alone and predicting what this profile would look like. Now we do this kind of predictions across the entire genome, and we really evaluate the global performance in terms of the correspondence and the concordance between the observed and predicted profiles. We see something really cool. This is the Jensen-Shannon divergence between the observed and predicted profiles. It's a measure of similarity of the shape of the profiles, okay? Since these are probability distributions, you can compute a distance between them, the KL divergence between the probability distributions, and you can symmetrize them. So you can compute the KL divergence of observed against predicted and predicted against observed. The KL divergence is not symmetric, right? And then you just take the average over both symmetries, and that's called the Jensen-Shannon divergence, okay? So it's simply a distance between the 2 probability distributions, one the observed, one is the predicted. And what we're seeing here is on the x-axis different resolutions. So you can compare the two profiles at single base pair resolution, or you can smooth them. You can smooth at 2 base pairs, you can smooth at 5 base pairs, you can smooth at 10 base pairs. And what smoothing will do is try to remove some sparsity and noise, right? Sometimes the observed data is very sparse. And so by smoothing it, you're kind of removing some noise. And the goal is to make predictions at different resolutions by smoothing the predictions also. And so you can really compare these profiles at single base pair resolution or 2 base pair smoothing or 5 base pair smoothing or 10 base pair smoothing, right? So the red dots that you see here are the actual comparison of the predicted profiles against the observed profiles, okay? At 1 base pair, 2 base pair, 5 base pair, 10 base pair smoothing. And so how do we know how good this is, right? These are just numbers, like you're getting some number like, 0.1, minus 0.1 or whatever. Is this good or is this bad? Well, this is a normal.
A quick question.
Yes.
So this is purely using DNA sequence. There is no information about chromatin accessibility, about histone modification. None of that is there, right?
No, it's just
is the conclusion that this entire binding profile depends only on DNA sequence, and doesn't it vary from cell to cell?
Yes. So this is a conditional model. So the way to think about this is you condition on a cell type, and condition on a cell type, the binding profile depends entirely on sequence. Now, when you go to a different cell type, you get a different model, and that will learn a different sequence profile, right? So we're not trying to make predictions in other cell types. We're just trying to see if you condition on a cell type on the trans environment of a cell, and you mutated sequences, for example, right? Or you generated synthetic sequences, or you didn't have a chromosome, right? How would the model make predictions on those unseen sequences in the exact same cell type, right? So in some sense, this prediction problem is useless because you already have the data for the other chromosomes, but this becomes useful when you start making synthetic mutations to the sequence, right? So the goal of this model is not so much a prediction, right? It's predicting effects of mutations in sequences. So the idea is that if you can make accurate predictions on sequences you've never seen before in training in that same cell type, then you could potentially, make any synthetic sequence you like and predict what the binding profile would have looked like in that cell type for that sequence, right? So this is not a cross cell type prediction model, and this is not claiming that claim would be absolutely wrong that like sequence is all that matters. In the grand scheme of things, yes, sequence is all that matters, but this is a conditional model where you are anchoring on a specific cell type, and your goal is to only condition on that cell type. How well does sequence predict binding, right? That's basically the goal of the model.
But your data is only 2,000 bases long at every point, right?
Correct.
So doesn't this mean that if you take a 2,000 bases long region and put it elsewhere in the chromatin somewhere else, it will behave the same, but it won't, right? Even in the same cell type, if you insert it in the heterochromatin,
yes,
you didn't get this pattern, no?
So it depends on the protein, right? So OCT4 and SOX2, I mean, that's one of the reasons we are modeling 2,000 base pairs, because if you model very short sequences, you actually get that kind of effect. Mm-hmm. But if you model like, every base pair in the model is actually getting to see what's happening in 2,000 base pairs away. And you could increase this input sequence. You could make it 5,000, 10,000, 100,000. So it gets to see the full genomic context. So if you move the sequence somewhere else, if you move the exact same 2,000 base pair sequence somewhere else, this model would make the same prediction, right?
Correct.
But if you moved 100 base pair sequence, it would have it would be context aware.
So this 2,000 bases, if you vary the distance, do you see large differences in your prediction, or do they remain, like if you make it 5,000 or 1,000? Does your
So 2,000 in the paper we show that 2,000 is like the optimal. So if you there are big differences if you model like 100 versus 2,000. Beyond 2,000, like even if you go to 10,000, there isn't a big impact.
I see.
So it seems like at least transcription factor binding and DNA's accessibility is much more local model. In fact, there are these like massive receptor field models like Informer from DeepMind. And I'll show you in a few slides, actually, our models beat Informer on many, many tasks. So the idea that you need to see like massive context is actually not entirely necessary. The model does make mistakes. And for example, a good example you gave is heterochromatin, right? If sequence is not the only thing driving effects, and there is an external, like, something that's in trans, right? That is not in the sequence, that is actively repressing that region, then the model, of course, makes a mistake, right? But the current model is just trying to understand what sequence can contribute to binding, right? And what we see is, at least within peak regions, you can get extremely accurate predictions. Outside peak regions, the model does make mistakes. It still gets a pretty high, AUROC, AUPRC. In the paper, we have the numbers. It's like the chromosome-wide AUPRC is something around 0.7, right? So it's not perfect. It's not 0.99, right? So it's making mistakes somewhere. But just from using 2,000 base pair context, it's getting most of the genome right. It's getting a pretty large proportion of the genome right. If you condition on chromatin, you would easily push that up to 0.9, right? So, but then you lose a causality aspect of the model, as you'll see later. As the primary goal of the model is to interpret, how the sequence is how the model is able to use sequence to make these kinds of really accurate predictions in peak regions, right? In regions where you see binding. But these are all good questions. The claim is not that sequence can predict binding across the entire genome, even in a single cell type. The claim is that 2,000 base pairs of sequence seems to be more than sufficient to get predictions on peak regions at base pair resolution extremely accurately. And outside peak regions, it still does really well, but it makes mistakes every now and then when the local sequence isn't sufficient to predict binding or no binding. So that can be overridden, right? Hope that makes sense.
I had one more question.
Yes.
since you care more about the DNA sequence itself, let's say you don't care about the chromatin, would it make more sense to combine the peaks across many different cell types to get like a theoretically where all coded bind, and then you can potentially integrate it with which region of the chromatin is closed and open rather than focusing on a single cell type?
That's not a bad idea. That's if you want to learn kind of generic rules of binding. But what actually we observe is that transcription factors have very different syntax in different cell types because they are, they are acting cooperatively with different factors. You can completely change, even though you're the same transcription factor, you're collaborating and cooperating with very different partners. So we really want to understand the cell type specific syntax. And this becomes particularly relevant when you're trying to understand effects of variants or mutations in very specific contexts, right? So the way we think of this is that the model is an abstraction of the experiment, right? So it's taking a ChIP-seq or a TAC-seq experiment, and you're learning a in silico abstraction of that experiment, where now you're trying to explain as much of that experiment as possible from DNA sequence. And right now it's a local DNA sequence model. We are starting to move into much larger receptor fields, but so far for attack in DNAs, we do not see any dramatic improvements by going to like 100 kilobases or 1 megabase. But obviously there is the prior history of the cell, right? That's what the chromatin is often capturing. Like the cell didn't just pop out of nowhere. And so if you really want to predict, binding perfectly accurately, you would need to know the past history of the cell, right? Like which is effectively encoded in the chromatin.
I just had a follow-up to that.
Yes.
So, but then this would mean, say you have an immune cell or something that gets activated, maybe then different factors come into play, right? And would the same cell that is trained on a resting cell work on an active cell then?
No, it would not. The goal here is not to transfer representations between cell types. I'll show you what the use of this model actually is. The goal of this model is to really understand the cell type specific syntax. So in that specific experiment, explain how sequence is driving binding, right? Or accessibility. And that's going to change cell type by cell type. And you can generate one model for hundreds of cell types. That's what we've been doing now. We'll be releasing these in the next year or two, next few months. And so you can really take an enhancer and you can track how sequence is dynamically, in fact, I'll show you, like you can do this for a sequence for different transcription factors as well, how the same sequence is being differentially utilized by different TFs, right? The same idea applies. You can take the same sequence and see how it is differentially utilized in different cell types. So you can have an enhancer that's active in, resting state and active T cells. And, there is a peak at that position. That peak is not often regulated exactly the same way. There are different motifs kicking in and so forth. So you can decipher the context specific syntax of that element. So again, the goal of this model is not to, it's not a universal model for OCT4, right? It's a model for OCT4 binding in embryonic stem cells. If you took a different cell type or you took a different time point or condition, you would have to generate the experiment and then train the model on that experiment and then do stuff with it, right? I'll show you what stuff we can do with it. Does it make sense? It'll become clearer once we see the utilization of the model. As I said, the prediction application here is completely like pointless because you already have, this particular cross-validation prediction objective is just to test if the model can make accurate predictions on sequences it has not seen in training, right? And these sequences that we are currently predicting on are sequences on held out chromosomes. But conditioned on the fact that now the model is very good at making predictions on sequences it has not seen in training, you can start using it to do all kinds of interesting experiments by mutating sequences that are in the genome and making predictions what would happen. And you can use that to study syntax and whatnot. Okay, I'll show you in a few slides. So as I said before, the prediction component of the model is less interesting. And I think what we are more interested in is how is this model able to predict this specific kind of spike in this enhancer for OCT4. And the same model, when you go to a different enhancer, says, oh, you know, the model is kind of fuzzy. I mean, the prediction is fuzzy, exactly like what you see in the data, right? And just to be, you know, I didn't finish this right here, but the performance of this model is on par with replicate experiments in terms of if you condition on peak regions, right? And you look at the predictions of the model, the shape of the predictions, you compare it to the observed data, you can see that the red and the blue curves, the blue are replicate experiments. The red curves are the actual predicted versus observed. They are very much on par and pretty far away from any kind of baselines, the average baseline, you know, random baseline. So the model is really learning the profiles extremely accurately at single base pair resolution. Okay. So we want to ask the model many questions. It's somehow able to map these sequences to profiles very accurately. How is it able to do that? We want to ask questions like which nucleotides in a specific input sequence, you know, for example, for this enhancer where OCT4, SOX2, and MANOG and KLF4 bind, what nucleotides are driving these effects? So there are two approaches we can take. You know, we are not going to try to interpret the parameters of the model. Some people do that. We think it's a very bad idea because these neural networks inherently learn distributed representations, which means that you know, no parameter in this model is remotely stable. Most parameters aren't even being used, and so, but that is the power of the model. It's like it's the fact that you have these distributed representations that gives the model the capacity to learn, the ability to learn very flexibly. But you should never overinterpret the specific parameters of any convolutional filter or anything like that. Even though they resemble motifs, there are people who look at these filters and actually think that they are motifs. They're not. I mean, you see the same kind of various versions of this filter in, like, you know, 30 filters will have the same kind of pattern, but slight modifications. None of that is actually, in quotes, real. It's just sort of the neural network, you know, distributing the representation across many neurons, and then they're all ensembling together, right? So it's dangerous to interpret the individual parameters of the network, but you can use the model as an oracle and like do in silico mutagenesis, right? So you can, if your goal is to understand what is the importance of this base in the sequence, you can literally just do a brute force approach where you mutate that sequence, right, that base to every other possible base, and just, you know, do this for every base, right? Saturated mutagenesis. And you can get an effect size for every base pair, right? For every possible transition. This is very inefficient because you have to basically do every base propagate forward 4 times, right? So there is a more efficient approach, a backpropagation based method. And this uses, you know, an idea similar to how you train the models with gradient descent, where basically the idea is that there you're trying to optimize the parameters of the model and using gradients to kind of get the optimal direction. But if you think about what gradients are, partial derivatives, a partial derivative of the output prediction with respect to any input base in the sequence, right, is telling you the infinitesimal change in the output if you made an infinitesimal change to the input, right? So it's effectively a measure of sensitivity. So if you're able to, you know, very efficiently calculate the gradient of the output with respect to every base in the input, that gradient value, right, would in some way give you like a sensitivity metric, right? It would tell you the contribution, the partial derivative contribution of that base to that output. Now, we had talked about, you know, the chain rule, right? So if your goal is to compute the gradient of the output with respect to this base, you can, you know, compute the local gradient and then start using, you know, classic chain rule to very rapidly and efficiently in one backward pass through the model, you can get the local gradients, right, of each of these these neurons with respect to the output. You can then get other local gradients, and using chain rule, you can compute the gradient of this with respect to each of these, and you can recursively backpropagate through the model, and you can, in one backward pass, you can get the gradients of the output with respect to every input base, right? It's a very efficient algorithm. And this has been shown before, but we developed a version of this. There are issues with using actual gradients. There's saturation issues through nonlinearities. So I'm not going to spend too much time on that, but we published DeepLift in 2017, which is a slight modification to the gradient algorithm. Instead of using infinitesimal gradients, it uses finite differences. Okay? And that there are other little tricks, bells and whistles, but effectively, it's the same idea. It's an efficient backpropagation algorithm that can take the output prediction of a model and one backward pass instead of, tons of forward passes, which is the brute force approach. It can give you in one backward pass the importance scores or the contribution scores of every base in the input sequence. So when we do that, so here's an enhancer, the actual, this is the OCT4 distal enhancer in mouse embryonic stem cells. Here's OCT4, SOX2, NANOG, and KLF binding patterns. And we're getting the same sequence. There are 4 different models, one for each transcription factor. And the same sequence is being interpreted through the 4 models. So we get these kinds of contribution scores. The height of the nucleotide in each position is giving you the contribution of that nucleotide to the output profile. Okay. And so you can see that it's pretty nice. It kind of, silences most of the sequence and then just highlights specific units, right? And if you looked at motifs long enough, you kind of probably see what you're getting at. These are classical OXOX motifs. There's a ZIG3 motif, there's a KLF4 motif. But you can see kind of this little TG80 sequence pops up specifically only in the NANOG task. This KLF4 really pops up very strongly here. The OXOX motif is common to all of them. So you can really start seeing context-specific base pair contributions. The same sequence is sort of utilized differently by different transcription factors. This is well known, but it's very nice to kind of see that a neural network agnostically picks that up, right? And it can give you this very context-specific prediction, which is not easily possible by just taking motifs and scanning, because all these motifs are present in the sequence. The model is telling you which of those motifs and which base pairs are being used for specific transcription factors, right? This is for one enhancer. So you can do this for every enhancer in the genome, right? Or every binding site for all 4 transcription factors in the genome. You can get these really beautiful context-specific base resolution contribution scores. Now you might want to say, okay, this is for one sequence, but I want to get I want to have a motif discovery algorithm that can, walk through all the tens of thousands of binding sites and summarize all the contribution, high contribution patterns it's seen. So we have this algorithm called Morisco, and what Morisco does is kind of builds off DeepLift. Okay, so what DeepLift does, as I mentioned, is it will take every sequence. You take a model that is pre-trained. Let's say for OCT4, you'll use that model to make a prediction for every sequence. So let's say there are 3 sequences in the genome that OCT4 binds to. You'll make predictions. You'll use DeepLift to backpropagate that prediction and get base resolution contribution scores. It'll highlight kind of different pieces of the sequence. Then what we do is we'll just filter the sequence. So we'll throw away bases that the model claims are not important or don't have high contribution. And you're left with these things which we call seclets. These are subsequences, but they are not classical subsequences because they have weights on every base. And then we have a clustering algorithm that takes these sequences, computes a novel distance matrix called a continuous Jacquard, which computes the distance between all of these little seclets post alignment. So for all possible alignments, we find the best possible alignment with the best score. And we do this for all of them. And we use a community detection method to cluster these sequences alongside figuring out optimal alignment. And then we collapse them. Okay, so we can get basically aggregate patterns, sort of average patterns of aligned high importance sequences with weights. So these are not classic position frequency matrices. They're not just frequencies, they actually have contribution scores. So we call these contribution weight matrices because they look like the position frequency matrices, but they're not. The values are actually capturing predictive contributions of those bases. Okay, and in the paper,
what does the negative contribution mean? It means if you mutate it to something else, it would improve. I mean, it is actually hurting.
Yeah, exactly. So a negative value would mean that you don't like that base, right? So if you don't prefer that base at that position. Yeah. So if you mutated it to that base, to that particular base, it would have a negative impact on binding. Okay. So when we do this, for all these 4 proteins, you actually pick up a really quite beautiful repertoire of motifs. Many of them pretty well known. There's the OXOX heterodimer, there's the OCT4 monomer, there's OCT homodimers, there's SOX2. I'll get to this in a second. We find kind of this was pretty interesting. We found a few different kinds of nanog motifs. There's KLF4 dimers. There's also indirect binding. So there's motifs or transcription factors that are neither OX, SOX, nanog, or KLF4. We find like ZIG3, SRRB, BBOX. So the neural network is sort of learning, the entire lexicon of monomers and homodimers, heterodimers, all of those kinds of things. And we pick up like 50 different motifs, which have all kinds of interesting variations, right? I'm just showing you the top few. And the nice thing is you can get the actual contribution of these motifs for all of the 4 tasks, right? So these are the same motifs. Some of these motifs are learned in all 4 all 4 models, all 4 transcription factors. But you can see like OX-SOX is really important for OX4 binding, SOX2 binding, nanog, and to some extent KLF4. This nanog motif is specifically, very important only for nanog. This KLF4 motif is super important for KLF4 only. And then you have BBOX, which is specifically relevant only for OC4. So you can capture these kind of, context-dependent effects of these motifs. And of course, we can get, these chip exo footprints from predictions. And you'll see that the direct binding motifs, the motifs create really sharp footprints when they are recruiting the proteins directly. So for example, the OX-SOX motif shows really sharp footprints for OC4 and SOX2. You can see again, nanog shows really sharp footprints for the nanog motif, but you see nanog shows kind of fuzzy, hazy footprints when it's indirectly recruited by OC4, SOX2, right? So there's very nice correspondence between, the sharpness, the dispersed nature, and you can see that the different motifs actually are driving kind of different kinds of chip exo footprints. So they're kind of nice way to show that these are not just like clustering artifacts. They in fact drive pretty different kinds of footprints. Okay. In the interest of time, I'll just skip this. You can check out the paper, but we did find kind of interesting variations of the nanog motifs. Some of them have nice validation from previous crystal structure experiments of the nanog protein bound to DNA. So I really want to focus on this piece now, which is because this is all, so far pretty standard. Like we found motif instances, we found motifs. Okay, we found some cool motifs, but this is where the models get really interesting. So one of the things that I mentioned is these models can not only learn motifs, but in principle, they can learn syntax, like all these other higher order properties of interaction effects, effects of spacing, orientation, all of that. And so we decided to use a model to really try to dissect those pieces of finding. And we try to do this in a way that, like, when a biologist is doing an experiment, it's like a black box. They don't know the system, right? So they're kind of like perturbing the sequence and then shoving them, like, think of like a reporter assay, right? You're taking sequences in the genome, you're deleting a motif, you're deleting another motif, you're deleting pairs of motifs. You create a library of mutations, you push it, give it to the cell is a black box, and the cell doesn't make predictions, it generates data, and then you take all that data and you try to infer the logic, right? So we did the same thing. We basically said, we now have an in silico oracle, which is like some abstraction of that experiment. Let's just pretend like we're doing, synthetic biology. So we can, for example, generate synthetic sequences. We can like place motifs, at specific positions, and we can just change spacing, right? So we keep everything else constant and we only look at spacing effects. Or we can take actual genomic sequences, real enhancers in the genome, and we can take, we've identified where the motifs are, and we can start making mutations, combinatorial mutations, and see what the model predicts and look for systematic patterns. So I'll show you two examples here. This is, we wanted to study the OX-SOX motif and the nanog motif are very often together in enhancers, right? Everyone knows that. We've seen it. Previously, people have seen it. But we wondered if there were any like interesting spacing constraints between these, right? There's been lots of interesting hypotheses about like a billboard model or a, enhanceosome model or a soft syntax model. What is a neural network learning and what is it saying about syntax? So the nice thing about this synthetic experiment is it's a very marginalized, very pure experiment, right? Like we take randomized DNA, we take actual enhancers, we just completely shuffle, randomize them. We push them to the model and make sure that they produce no activity. And then we insert a clean nanog motif at one position we insert an OXOX motif at another position. And then all we do is we create a library of sequences where we're keeping nanog's position the same, and we're just moving OCT4 SOX2 motif towards a nanog motif. And here's sort of the animation of what's happening. The model's making predictions as we're moving. This is the motif distance, and you're moving closer, and you're seeing the model like respond to the spacing, right? And if you look carefully, you'll see kind of nanog bobbling, right? You'll see kind of like initially it's not really responding, and as it gets closer, you kind of see this kind of bobble effect, and then it gets really strong, right? So if you graph the response of nanog and OCT4 as a function of spacing, you get something that looks like this, which is really interesting. The red curve is the response of OCT4 to just the spacing syntax. And you can see that like OCT4 literally doesn't care. You can change the spacing quite a bit, and until you get really close, basically there is almost no response. Like the fold change in the footprint height of the signal is very, very marginal, right? But just see what happens to nanog. So what happens with nanog is like, as you decrease the spacing between OXOX and nanog, you see this kind of exponential rise in nanog's binding, which is really interesting, indicating that nanog is responding to that spacing between OCT4 and SOX2 and the nanog motif. But what's even more interesting is as you get closer, you start seeing a periodicity. And this periodicity is actually exactly multiples of 10.5. So what it's saying is that there's a cooperative effect. It's unidirectional. Basically, I mean, so it's kind of makes sense largely. OCT4 is a pioneer factor. It binds the OXOX motif. It doesn't care what nanog's doing. Nanog is heavily responding. It is binding cooperatively with OCT4. And it's a distance, it's a soft syntax, it's a soft distance dependent cooperative. It's not like there's a specific spacing that only works. There's an exponential kind of cooperative effect. And then there's also built-in periodicity. So when the OXOX motif and the nanog motif are on the same side of the helix, there's cooperativity. When they're opposite side, you literally see the opposite, right? You see a massive, you go from 10.5 to, the next iteration, right? The next opposite side of the groove, and you see a complete loss of binding, and then you gain again, loss, again, loss. And so it's distance dependent with the periodic frequency, and it is directional, right? So this just pops out of the model. It's really quite beautiful. Now we can repeat this in silico, in the genome itself. So you're taking that same OCT4 enhancer, and I'm going to repeat this now in the actual enhancer. This is not a synthetic sequence anymore. The sequence contains the OXOX motif and a little nanog motif. And as I showed you before, OCT4 doesn't seem to care about the nanog motif. The nanog binding seems to care about the nanog and the OXOX motif. So I can delete the OXOX motif and predict the effect on OCT4 binding, and it predicts a complete loss of OCT4 binding. And if you delete the OXOX motif, keeping the nanog motif intact, you see what happens. The nanog binding also completely gets destroyed. Even though the nanog motif is intact, it's completely gone, which indicates that there's almost complete dependence on nanog on OCT4, OXOX motif being present and OCT4 binding. If you do the opposite, where you delete the nanog motif, OCT4 doesn't care. And actually, even though you deleted the nanog motif, you still see kind of a weak footprint for nanog, indicating that the OXOX motif can completely indirectly recruit nanog. It doesn't require the nanog motif to be next door. Okay. And if we now do this for all enhancers in the genome, and we kind of plot again this distance curve, but this time using actual enhancers, you see something very similar, that again, OCT4 doesn't react to the spacing, except until you get really close and you form an OXOX nanog, tetramer, a classic, composite motif. But again, you see nanog kind of responding, and again, you see the periodicity, right? It's weaker. That's because enhancers are not pure. We are just looking at like the pairwise effect of two motifs. There are other motifs going on which are having cooperative effects. The previous simulation is completely marginalized. So it gives you extremely pure effects of just OCT4 and nanog again. So we learn all of these kind of really interesting pairwise relationships in the paper. We have a lot more other stuff, all kinds of spacing dependencies. This periodicity is very specific to nanog, by the way. We don't see it for everything else. But there's all kinds of interesting distance-based, there's direct, very short range cooperativity, and then there is nucleosome range, like up to 150 base pairs. There's actually cooperative effects. That's probably chromatin-mediated cooperativity. So I've run out of time, so I'm just going to show you one last thing and then stop. I guess we can get time to talk about the genetic variants. But we did validate a bunch of this for the paper. So Julia's lab actually did some cool experiments where we did actual CRISPR experiments with motif mutations to test, how the model's predictions validate against the actual experiment. So here we're seeing this is the observed profile for some enhancer. This is the predicted profile for the same enhancer. Here the model predicts a SOX2 motif, and the model predicts that if you mutate these two T's and you make them into AGs, you'll have a specific kind of effect. And this is the predicted effect. You see that the model predicts a loss of footprinting here and also an attenuation of the footprint reasonably far away, right? It's not super far away, but it's like, some 80, 60, 70 base pairs away. So it is pretty long distance, right? For SOX2 itself. So if you actually do the experiment, that's what you see. You see loss of the footprint here, and you see an attenuation here, right? Now, what happens is you can now do the exact same experiment, the same CRISPR experiment, but now you're chipping NANOG. So you're looking at NANOG's binding profile. And this is the prediction of the model that when you mutate the SOX2 motif, you can see a pretty decent effect on NANOG, and it's pretty widespread. You can see effect sizes pretty far away, up to like 150 base pairs away. When you perform the experiment, that's pretty much what you see. You see a long distance effect of mutating SOX2 on NANOG, right? Now you can invert that. There is a NANOG motif right here. And if you mutate this NANOG motif and now you measure NANOG binding, you see kind of a mostly proximal effect. It's again, not super proximal. It can extend reasonably out. This is what you see with the actual experiment. And now if you do the same mutation, but you measure SOX2 binding, the model predicts no effect on SOX2 and the experiment shows no effect on SOX2. So this is kind of verifying the directional cooperativity with some of the distance-based constraints that the model want, right? We had a few other validation experiments in these. These are super difficult an experiment to do. So we just had 3 or 4 these kind of point mutation CRISPR experiments. But hopefully that shows you, that the model can learn this kind of beautiful syntax. And now we've been applying this to hundreds of different transcription factors and thousands of cell types and trying to derive all these higher order syntactic rules. And there's incredible, distance-based cooperativity. We don't see lots of evidence for, the classic enhanceosome model. I think those are exceptions to the rule. There are obviously composite motifs, transcription factors that form either protein-protein interactions or bind jointly to very closely spaced composite motifs. But there's a lot of medium-range cooperative in nucleosome range cooperativity that seems to be soft syntax. It's all like, exponential decays of different rates. Some of them have periodicity. Some of them are other kinds of interesting structures, but they're all soft. It's not like there's only one position 80 base pairs away that works. Most of those seem to be transposable elements, but that's another interesting point we make in the paper, that a lot of the previous, fixed syntax evidence that people have shown before appears to be confounding with actual transposable elements. At least that's what we find in this paper. Okay. I'll skip this, but you can imagine in your head, maybe I'll just show you one really quick thing, is you can look at mutations, actual genetic variants, and you can, take the model, you can insert a reference allele, the model makes a prediction. You can switch it to the alternate allele, the model makes a prediction, and you can see this kind of beautiful allelic effect. And you can also explain how that variant is creating that allelic effect, right? The C to G transition creates this beautiful, strong motif that drives binding or accessibility. Anyway, we have some cool results and the papers. We applied it to Alzheimer's disease in a Nature Genetics paper. We've applied it to also rare diseases. So congenital heart disease using single cell ATAC-seq data from fetal hearts. We have some really cool validation of one of the predictions in a real patient, from a real patient's genome. We see very nice CRISPR, validation of the model. We see luciferase validation, and we even see phenotypic effects of the mutation in arterial endothelial cells. So, the models are pretty good. They can not only give you kind of really interesting insights into the syntax of regulatory DNA, but they can also be used to study in silico effects of common and rare disease genetic variants and mutations. Okay. Sorry, my slides were. I'm glad you asked questions and we spent some time discussing things. So I unfortunately couldn't get through the second half, but hopefully it gives you a picture of what we can do. So I'll just stop there and thank my lab. This project was specifically led by Ziga Abjek, who was actually a summer student who visited the lab from Germany. And he was amazing. He spent 3 months with us in person, and then 2 years after going back was grad studies. He's now at DeepMind doing kind of similar things. The Informer model is in fact from him and his group. But this is my lab. We've been applying these kinds of models to lots of different kinds of data and different kinds of genetic variation, different kinds of diseases. And none of this is possible without our collaborators. So we are a pure wet lab, dry lab. We primarily do almost exclusively computational work. So we rely heavily on a network of collaborators and of course, our funding sources.
Thanks.
So let me know if you have any more questions.
Very nice talk. We might have time for one quick question if there is.
Mm-hmm.
One? So I have one quick, it will really be quick. So when you were doing the mutations, you said directional differences. So did you mean that if you invert the motif, you would not, reverse the motif? You would not.
What I meant directionally is it's not symmetric cooperativity. That's what I meant in this case.
You mean left, right? In that.
Yeah, I just mean that that OCT4 and NANOG are binding together, and the direction of cooperativity between OCT4 and NANOG is asymmetric, right? It's like NANOG depends on OCT4, but OCT4 doesn't depend on NANOG. That's what I meant by symmetry.
I see. But you must have tried inverting too, right?
Yes, in the paper we have like all kinds of other things. We have inversions. We have you can change the affinity of the motifs too, right? So you can do all kinds of cool things. You can, in fact, we have a follow-up paper where we show, in fact, that we can extract thermodynamic binding energies from the neural networks ab initio. That's really exciting because, all of this kind of is interesting, right? You're having some feature importance and blah blah. The question really is is the model actually learning physics, right? Is it learning thermodynamics? And in fact, this affinity distillation paper that's on bioRxiv, we show that this exact same model I just showed you, this BPNet model, we have a way of extracting what we think is affinity for all kinds of k-mers. And if we perform in vitro experiments where we, like CLEX or PBM or BETseq, and we actually measure binding energies, we get amazing correlations. And in fact, we can just calibrate the models in the range of 0.8 to 0.9 across a very wide dynamic range. Not just mutations in the motif, but also mutations in the flank and all kinds of stuff. So there's nice evidence now that these neural networks are actually also indirectly learning affinity and thermodynamics.
And do you get these very nice correlations even with ATAC-seq?
Yes, in fact, we have the preprint isn't out yet, so we have a similar model called Chrome BPNet. And that model is also extremely accurate. It's the same. It's like replicate concordance for profile shape. There's a pretty important component to it, which is the enzyme bias. So we have an enzyme bias correction approach, but that model, in fact, I didn't get time to show this, but we use that model to, for example, score molecular QTLs, and we outperform much bigger models like transformer-based models and so forth.
All right. Thank you. I'm just sorry. I would have liked to ask more questions, but we're just completely out of time.
Yeah, no problem.
Thanks, Anshul.